future for sure.
Yeah.
Okay, we are online. Good afternoon, everybody. We are here again for one more SBI talks. I'd like to thank everybody that is online, and I'll talk something in Portuguese for the students. So, estudantes ou algumas pessoas que estiverem online no site, no Facebook da SBI. Nós vamos começar a transmissão com a doutora Amanda e com o doutor Dario. Fiquem à vontade para escrever as perguntas no chat em português, que eu traduzo para a doutora Amanda, se for necessário, ok? So, Amanda, thank you very much for accepting our invitation. It's a pleasure to have you here. We are beginning our meeting. So, Amanda is professor of molecular microbiology and immunology from Brown University at the United States. And she's working on some data about COVID-19 and lung epithelium. So she's going to talk about the role of the lung epithelium in directing COVID-19 induced pathology. Amanda, we appreciate your presence, your virtual online presence here, and we hope in the next future you can be here in Brazil. And so you can begin your talk. Thank you very much.
So thank you very much. I'm just gonna share my screen. There we go. Sort of gotten pretty good at this Zoom thing recently. And of course I would thank you very much for the invitation, and been to Brazil a couple times now, and of course love to come back. So today I'm going to talk about some really recently sort of derived data that's unpublished. Some of it's on bioRxiv, some of it hasn't even made it there yet, about the role of the lung epithelium in directing COVID-19 induced pathology. So my lab has been working on the concept of host disease tolerance for quite a while now. And what that idea is that if you're infected with a pathogen, you can either clear the pathogen, so that would be resistance, or you can tolerate the damage caused by the pathogen or the damage caused by the host response in some cases. And so this tolerance responses is really what we've been focused on. And so a susceptible host will either not clear the pathogen and or not tolerate the immune-mediated damage, or sometimes the pathogen-mediated damage or other sort of effects of the pathogen. And so this is another way of showing this. So in this case, this is host fitness on this axis and pathogen burden on this axis. And you can see that this is a much less tolerant host, because as the pathogen burden goes up, it becomes less fit, where this is a much more tolerant host. And so we've been looking at the concept of host tolerance to infection in the sort of looking at it in terms of pneumonia. And there's a lot of reasons for this. So pneumonia is the deadliest childhood disease, infectious disease. And as a lot of these infectious diseases have gone down over the years, pneumonia has also gone down, but not nearly as much as some of the other infectious diseases. And so one of the reasons is that it's such a prevalent pathogen is because it can be caused by bacterial, fungal, or viral infections. And oftentimes there's mixed infections. So viral followed by a bacterial infection or a fungal infection. And so this is what we've really been studying, is how viral infections predispose people to a secondary bacterial infection. And there's several reasons for this. It was thought in the 1918 influenza pandemic, there were 50 million deaths worldwide, and a secondary bacterial co-infection was confirmed in a majority of these cases. And now that we have antibiotics, of course, things are much better. But in 2000, even in 2009, and with the pandemic, co-infections were confirmed into about 55% of the fatal cases. So, and this pneumonia is starting to get the interest of the NIH, which is where I get a lot of my money. So it's good that they're interested in that. Here there's a lot of different things can happen to increase host susceptibility, including age, genetics, exposures, and acute conditions. And we're really focused on this host response, where there's immune resistance, microbiome changes, tissue resilience, repair and resolution pathways. And then what's really important to remember, and this has actually really been shown in COVID-19, is there's a long recovery period for pneumonia, and there's a lot of exacerbation of chronic diseases. And well, now we're hearing about this long COVID, where people really have sort of a lasting effect of lung infections. So our idea is that you want to return to homeostasis after these complex and severe lung infections. So you can do this. This is an alveolus here, sort of an outline of this, where you can have this transient damage, and then you return to homeostasis. But if you can't return to homeostasis, you're going to end up with this pathological damage. And this is where the long-term consequences come and the severe morbidity and mortality that can be caused by pneumonia. So this is a review that we published several years ago, really focusing on host tolerance mechanisms in pulmonary infections. And so these are several of the ways that we feel that you can increase host tolerance. And one thing that we're really focused on is the maintenance of the barrier function. So basically looking at epithelial and endothelial cells and types of cell death that happen there. You can also make sure that there's less inflammation, so anti-inflammatory cytokine secretion, early control of the infection. And then one thing that I worked on in my postdoc and we're continuing to work on now is really this initiation of repair and remodeling. So making sure that if there is damage, that it's properly repaired. And we're also looking at how the microbiota plays sort of a role in all of these things in this pulmonary tolerance to infectious disease. So what I initially was working on was looking at ways of decreasing inflammation and repairing damage. And so we found that there's a role for tissue protection in lethal respiratory viral bacterial co-infection. And this was published several years ago. And this basically summarizes that when you have flu infection and then you have a Legionella infection following that, you get a lot of damage, but you don't get an increase in pathogen burden. So if we can really focus on this host response, repair the damage, decrease inflammation, we can increase survival. And more recently, we've been working on ways what we found is that it depends really very much on the secondary infection, the bacterial infection that follows flu infection. And we've really been focusing on ways that alveolar macrophages and macrophages in general have a role in sort of promoting the repair processes as well as clearing the infection. And so this was published recently. So what we like to think in the lab, because we're really obsessed with the lung, I just wanted to actually point out that I was obsessed with the lung before we had this pandemic. And we really think that it's a central regulating node. And it's not only is important in controlling immune responses, of course, it's important in controlling blood flow as it works with the heart, respiration, metabolism. And what we're really starting to focus on, so my initial training was in innate immunity, but now we're really realizing that the lung epithelium is a really important part of the innate immune response and also a huge part of the host tolerance response. And so when we look at host tolerance, the advantage of this is we can have these host-directed therapeutics. And we can basically target the host response and instead of trying to figure out targeting each pathogen with vaccines or antivirals. And the advantage of this is they should be able to work across a wide variety of pathogens. And so what we want to try to figure out now is, can we apply this idea of host tolerance and host-directed therapeutics to SARS-CoV-2? But first, we really need to understand the pathology, and especially what I'm going to talk about today, the pathology as it relates to the lung epithelium. So I don't think SARS-CoV-2 needs any introduction. This is just showing how it spread from January 2020 to March 2020 to May 2020. And I think they sort of, I don't even know if this map exists anymore. I think they've given up because it would just be a big blob of colors at this point. And what we want to know are there dysregulated host tolerance pathways. So the SARS-CoV-2 disease profile is that it causes a severe pneumonia. It infects the lung epithelium. It can cause acute respiratory distress syndrome. There's some data suggesting it also causes a cytokine storm and acute lung damage, but then also systemic organ dysfunction in the very severe cases. So this is looking at SARS-CoV-2 complications and pathology. So we know that it causes this pneumonia and the systemic cytokine storm. But what's really starting to be clear in many, many case studies and larger papers now is that the pathology of COVID-19 is also associated with increased blood coagulation and clotting. And so this is one of the first papers that showed this, that there's a procoagulant pattern of patients with the COVID-19 acute respiratory distress syndrome. And this isn't unknown in ARDS or in ALI, that you can get these thrombosis and this coagulopathies, but it seems much more severe than in other viral lung infections. And so here's some more clinical reports showing that all this year where there's disseminated intravascular coagulation in about COVID-19 non-survivors, deep vein thrombosis in about 27% of ICU patients, pulmonary embolisms in about 22% of hospitalized COVID-19 patients, and then this can also lead to stroke in a smaller subset of COVID-19 patients. So there's many potential triggers of coagulopathy. So it's, as I said, it's seen in the acute respiratory distress syndrome, and it's thought to be caused by induction by tissue damage and systemic inflammation. So this is where the immune system comes in. If you have this really strong inflammatory response, you're going to get this activation. So this is the bronchial here, and you're going to get this activation of the from the neutrophils and these other immune cells coming in that will activate the sort of fibrin system. And in this way, it's probably trying to be protective. It's basically trying to repair damage that's happening. But if it gets too much in one direction, then it will decrease host tolerance responses and it will basically cause more damage. So we decided to look at some, well, the lab shut down. And so the people that do bioinformatics in my lab, this is in March, decided to start looking at publicly available data sets to see what we could find. And so we looked at this from this first data set that we looked at was from published with Blanco Mello et al. And it was looking at imbalanced host response to SARS-CoV-2 drives development of COVID-19. So they're really sort of focusing on the imbalanced immune response. So we took this data and looked at it in a slightly different way, and to look for different things such as the maybe coagulation. And so what they did in this is they basically infected primary, so healthy, normal human bronchial epithelial cells that were grown in culture, and they infected them with SARS-CoV-2. So you can see here, what we wanted to do was look at the overrepresentation analysis. So what genes were overrepresented in our analysis. And we found that platelet degranulation and the regulation of blood coagulation was differentially expressed. And so this is shown here. So these are a bunch of genes that are important in blood coagulation, and I'm going to go into these pathways, which are quite new to me as well, in a moment. But you can see that there's a strong upregulation of a lot of genes that are important in blood coagulation in the infected cells. So it was a bit surprising to us at first that the lung, we think the lung epithelium is important for everything, but we didn't realize that it was this important for possibly driving blood coagulation. And it really hadn't been discussed in the literature in terms of infection so much as an important driver of blood coagulation. So most of it is focused on the endothelium, which of course plays a really important role in blood coagulation and also on immune cells. But if you look through the literature, we found some papers where they talked about the role of the coagulation cascade proteases in lung repair, that the alveolar epithelium can initiate extrinsic coagulation cascade through expression of tissue factor. Alveolar procoagulant activity is important in recovering from acute lung injury by lung epithelial tissue factor, and tissue factor signals airway epithelial basal cell survival in this sort of in vitro study. So it may have a protective role in acute lung injury, but then there's these pathological consequences. So this is a brief primer on the regulation of coagulation. So these are the genes that I'm going to be talking about for the remainder of the talk. So this is looking at the plasminogen activator inhibitor 2. And this is looking at urokinase receptor plow. So this whole plasminogen activation system is important for understanding what happens in during the infection. So what happens first, you get this extrinsic coagulation cascade, which the main factor here is tissue factor. All these genes are sort of have a bunch of different names, which makes this even more confusing. But so it's also referred to as F3, so TF or F3. And then a really important inhibitor of this pathway is the tissue factor pathway inhibitor, TFPI. And so you can see in red, these are all the things that inhibit the extrinsic coagulation cascade. But if these are not inhibited, it will form this cross-linked fibrin clot. So the plasminogen activation system is actually really important in getting down to plasmin, and plasmin is really important in actually inhibiting the fibrin clot and breaking it apart. And so these are all then this is all tightly regulated by the plasminogen activator 2, urokinase receptor, urokinase, and tissue plasminogen activator plat. So these sort of are the inhibitory side, and these are some inhibitory sides, and this is the main activation pathway of coagulation. So if you look back at the data with the SARS-CoV-2, you can see clearly that there's, if you look especially at F3, so this is the tissue factor, so this is the main activator of coagulation, and you can see that this is highly upregulated in the SARS-CoV-2 infected cells. And then some of the genes that are important in inhibiting are downregulated. I'm gonna sort of go through these, sort of highlight some of the important genes here. So here you can see that this tissue factor F3 is highly upregulated in SARS-CoV-2 infected lung epithelial cells. And TFPI doesn't seem to really be impacted at all, at least it's not statistically significant. So that's this inhibitor of the pathway here. Whereas PROS1, which is another inhibitor, is actually downregulated. So this basically means that everything that's triggering the activation of coagulation is being upregulated, and then the inhibitors are either staying the same or being actively downregulated. And so this really demonstrates that the lung epithelium could really be playing a role in this coagulation pathology. So then if you look at the plasminogen activator side of things, so here you can see that the plasminogen activator is upregulated. The urokinase receptor plow is also upregulated. So a lot of these things are upregulated, but also the inhibitors are upregulated. And so basically you're having both inhibitors and activators upregulated, which could lead to a defect in plasmin production, which has been seen in patients. And so this could also lead, the fact that this important regulator of this fibrin clot is not being properly regulated could then also lead to these coagulopathies. So then we wanted to know, because the data from the clinical data was suggesting that this SARS-CoV-2 pathology, this coagulopathy, is much more prevalent in SARS-CoV-2 than it is in other viral infections. So we wanted to know, is this specific for SARS-CoV-2 infected lung epithelial cells? So here we looked at so normal human bronchial epithelial cells infected with SARS-CoV-2, or those same cells infected with flu. And so this is back to the Blanco Mello data that they had this data in that paper as well. And so when we reanalyzed that data, we found that you can see here that the flu does not have that same really stark sort of expression patterns that's seen with the SARS-CoV-2. And if you look at things like F3, it's not highly upregulated compared to control, whereas with SARS-CoV-2, it is. And so this is the tissue factor, the gene that's really important in starting off the whole coagulation pathway. So then we wanted to know if these findings that were from these in vitro assays held true in patients. So what we're seeing in this data from submerged culture, also important in SARS-CoV-2 patients. So we went to look for, and so this a few months had gone by, and so now there was more data available. And so we looked at publicly available patient RNA sequencing data from the Zhang'an Hospital at Wuhan. And this had 3 severe COVID-19 patients and 3 healthy controls. And so they did bulk RNA sequencing of RNA cells from the bronchial lavage fluid. So that's the fluid that basically wash out the lung. And so this is going to be a complex mixture of immune and epithelial cells. And they also looked at peripheral blood mononuclear cells and with the same patient data set. So when we looked at peripheral blood mononuclear cells, you can see that there's really no significant pattern in gene expression when it comes to the coagulation, the genes that are important in coagulation. When we looked at the BALF from these same COVID-19 patients, you can see that there is this very distinct pattern of expression. And so what we saw in the BALF was actually strikingly similar to what we saw in just the lung epithelial cells, not identical, but quite similar. So we think this is because a lot of the cells in the BALF, especially in the infected patients, are going to be sort of sloughed off lung epithelial cells. And so a lot of the signal is going to be coming from that. However, what we really needed to do to sort of pin this down was to be able to do look at single cell RNA-seq. And so fortunately, someone published that as well. So we looked at bulk RNA-seq data from patients in BALF, and we found, and I'll show that data in a moment, but basically with single cell RNA-seq, they took the bulk BALF, they did single cell analysis, and then they found the expression on each cell type. And then you can kind of see what the expression pattern looks like and thousands of cells and subset those cells per type. So this was from this study here by Lorenzo Chao et al. And so what they had was these patients from Germany, where they had 19 COVID-19 ICU patients' nasal swabs and bronchial protected specimens, so where they put something down into the lung. And then they had 2 bowel samples from some severe patients who later died and 5 healthy controls from volunteers. And so this is just showing this data where these different, so based on sequencing, how the different cells subset. So all you need to really know for these purposes is, in this case, the lung epithelial cells are over here on the left. Neutrophils, macrophages, and a lot of the immune cells are up here. So this big blue part is neutrophils, and all this is different types of lung epithelial cells. So then when we looked at our favorite genes and the expression pattern of them, so this shows not only how many cells are expressing these genes, but how strongly they're expressing them. So you can see clearly here that that F3, this tissue factor, so the gene that really starts off the coagulation pathway, is really strongly expressed in many cells of epithelial cells, also a little bit in neutrophils and macrophages, as we'd expect. TFPI doesn't seem to be strongly upregulated in very many cells. And PROS1 is, again, really strongly upregulated in subsets of lung epithelial cells. So then when we looked on the other side, so that's the extrinsic coagulation pathway, but then we looked in the pathway, the plasminogen activation system, where we'd want to see sort of the inhibition of this. You can see that there's some expression in epithelial cells and also some expression in neutrophils. So this really backs up our data that a lot of the gene signatures that we're seeing in vitro recapitulate at a single cell level from patient data. And then really even more recently, so this patient from Lau et al, it was another single cell RNA-seq data set of COVID-19 patient BALF. And so this is data from the Shenzhen Third People's Hospital. And what's really interesting about this data is they actually did this, they stratified the patients by severity. And so in this case, the epithelial cells are here, and then these are different immune cell subsets over here. So if we just look at the epithelial cells, you can see really clearly that these genes, our favorite genes of choice that are part of the coagulation pathway, are, as you go by severity, some of the genes that are important in controlling the coagulation, to preventing it, are actually very strongly downregulated, while genes that are important in triggering it are strongly upregulated. And this is another way of looking at that data where it's stratified by disease severity. And here you have the sort of red in here. So the relative expression is more red. And then the percent of cells that are expressing it is a bigger dot. So this is really interesting because it allows you to see not only how much is being expressed by each cell that's expressing it, but the number of cells that are expressing it. And so this really backs up much of what we saw with the in vitro data. So this is just to summarize our findings from looking at reanalyzing these published data sets. And what we're hypothesizing at that virus-induced lytic cell death releases cellular contents, you get this lung-blood barrier integrity degrades. But what we're saying is that there might be epithelial-derived factors that drive coagulation, and then they will interact with endothelial cells and circulating cells. And you'll not only get localized clotting in the microvasculature from these COVID-19 patients, but because the lung is so central to the whole blood flow, you can get this distal COVID-19 coagulopathies, and the lung epithelium might be playing a role in basically this sort of whole body control of coagulation. So the path forward now, which is of course where we want to go with this, is bench to bedside. And Dario is actually going to help us with this. So what we want to do is we want to take our RNA-seq data, and we have a sort of list of small molecule drugs that we want to test. And we found this list based on the connectivity map database that the Broad Institute has. And test these in our in vitro systems. So we generally use an air-liquid interface model to look at lung bacterial co-infections, and now we're adapting that to SARS-CoV-2. And we're also using some different cell lines to sort of screen these small molecules and see if they have this similar gene signature, and if we can change that coagulopathy gene signature. So I just want to get back to the idea that if we study the lung, we can understand the body. And so this is really important, like regulator of much that's going on in the whole body. So with that, I just want to thank you for your attention, and I want to thank the lab. Some of the co-infection work was done by Kayla Lee Campbell, and so she's moved on and doing a postdoc now. I just got a nice grant to work on this from Emergent Ventures. Ethan Fitzgerald did all the bioinformatics data, and then these are other members of the lab. And I'd like to thank my department, collaborators. Here's what our lab picture looks like now. This is what it looked like last fall. And thank you very much for your attention.
Thank you very much, Amanda. Nice presentation. So we have some questions here. There's someone asking here, do you have an idea why there is such a difference between SARS-CoV-2 induced coagulopathy and influenza virus or other viruses? Why is it so different?
Yeah, I mean, we really don't have any idea. And so it seems to be something that the virus is doing specifically to the lung epithelium. And so why it's doing that, or is it some sort of severity metric? But we've gone back now and we've started looking at even very severe flu infections, and you just don't see it with the same frequency. So it's a bit of a mystery of what SARS-CoV-2 is doing to change the coagulation genes.
Yeah, and the data from severe patients is really interesting. So the infection with SARS-CoV-2 is really induces something on the according to the severity of the disease. It's very clear.
Yeah.
And I have another question here. Do you know, we know that some hormones like estrogens, they usually have a relationship with coagulation disorders. So, and otherwise we know that men usually have more severe disease, more COVID-19 with severe breathing support and
Right.
more adverse outcomes. So do you have an idea if this kind of regulation of endocrine immunoregulation affects this coagulation or this thrombosis induction?
That's a really interesting idea. Yeah, I don't think that's been, I mean, it's clear that men get a more severe disease, but why exactly, it's not entirely clear. So yeah, that would be really interesting to try to tease apart in the patient data, you know, look at some of these factors. And I'm not, I mean, I know men have worse pneumonia impacts. I'm not sure if there's data yet on if they have more coagulation defects as well, but that might be hard to tease apart because if you get more severe disease, then you have this coagulation.
Yeah,
so there's sort of a lot of factors to consider there. Yeah.
So in this data, you don't have this division, men and women.
You know, I could go back and look at it. Yeah, I don't know, and I would be concerned given the numbers if we tried to subset it further, that then some of the statistics would not be that strong.
Okay, Amanda, we have I will have to end your participation with one last question because of our time, if you don't mind, and then we proceed to Dario talking. There is another question here, just the last one. Do you know if the local microbiota can influence somehow this coagulopathy in lung, or is there any kind of expectation about that?
So we're just getting into whether or not, actually something that we're studying now is whether or not changes in lung microbiota or either lead to a secondary infection with bacteria. So there have been some reports of that. It's not, that data is still coming in, but does that change? So we're actually just now starting to look at lung microbiome changes with infections. So that would actually be a really interesting question to see if that also correlates with the coagulation defects, because I could see that, you know, triggering more damage or causing some of these changes. That would be really interesting. Yeah, there's a lot of factors to consider.
Yes.
It's a complex disease.
It is. So Amanda, thank you very much for your participation on our talk. And I will now invite Dario for his talking. So it was really a pleasure to have you here. Thank you.
Thank you.
Dario, it's up to you.
All right. Thank you so much, Chris. Thank you, SBI, for the invitation. It's a pleasure to be here and present our last data about the COVID-19 and inflammasome activation. So this is sort of unpublished data yet. We put it on Med Archives, but it's still in revision. So I'll be happy to present this data and discuss the recent data we have about this. And I would like to start with the question that motivates the research in our lab is how the cells from our body recognize microbes and distinguish these microbes between pathogenic and non-pathogenic microbes. And if you think about recognition of microbes, many people think about toll-like receptors. They are very important indeed, but they cannot tell if the microbe that is dealing with our body is pathogenic or not. So as soon as the pathogens goes inside the cells and start replicating, modulating the functions of the cell, secreting molecules via specialized secretion system, or replicating in specific compartments, or lysing the membranes, you name it, the specific pathogen, the process that the pathogen may manipulate, we're going to have intracellular receptors that can sense and somehow discriminate between these pathogenic microbes, which is the ones that can damage our cells and promote disease, versus the microbes that are non-pathogenic, which is going to be outside of the cells or inside the phagosomes, and is going to be degraded by the phagolysosome. So we pretty much focus the research in our lab intracellular receptors, which is the receptors that senses and can signal and induce inflammation in response to pathogenic microbes. And this is critical for our immunity and for generation of adaptive immunity and survival against many microbes that may infect our cells. And if you think about intracellular inflammasome receptors, you think about inflammasomes, and I'm not going to spend too much time in the interest of time in this conference, in this talk about inflammasome, but we have many different inflammasomes. I just depicted 3 of them. This is a very well-known and well-studied inflammasome, which is activated by flagellin from gram-positive or gram-negative bacteria. The flagellin is sensed by the Innate proteins, which signals and activates NLRP4, which has a CAR domain in the carboxy terminal region of the molecule, which binds to the carboxy terminal region of caspase 1 and activates these proteases. We also have AIM2 inflammasomes that senses double-stranded DNA, and they use this adapter protein ASC, which is a pyrene card, a small protein that the pyrene and pyrene binds in a homotypic interaction between AIM2 and ASC, and the CAR domain binds to CAR domain of caspase 1. And of course, we have also the most studied inflammasome so far, which is the NLRP3. It's still open the question of how exactly the mechanism of activation of NLRP3 occurs, but it's definitely something not dedicated to sensing microbe molecules. Instead, NLRP3 senses the ROS, efflux of potassium, and cell damage, which may be a result of many cellular processes and many infections. And then NLRP3 can be activated. As soon as the receptor is activated, there's going to be interaction of the purine domain of NLRP3 with the purine domain of ASC again, and ASC has this CAR domain, which is going to interact with caspase 1, leading to caspase 1 activation, and this is the canonical activation of the NLRP3 inflammasome. Recently, there was a beautiful paper by Hao Wu at Harvard showing by cryo-EM the structure of activation of this platform, the NLRP3 inflammasome. And what they show is that during activation of the NLRP3, there is a conformational changes in the MBD and in the carboxy terminal region of the protein, and therefore NLRP3 can oligomerize and form this beautiful ring-like structure that is composed by 11 folds, so 11 molecules of NLRP3, which can now triggers polymerization of ASC and activation of the NLRP3 inflammasome. So if you rotate 90 degrees this structure, you're going to see a ring like in form of disc, and in the middle of this ring, there is the CAR domain of or the purine domain of NLRP3, which is going to be important for polymerization of ASC and activation of the inflammasome and the proteins downstream of this molecule. So this picture somehow summarizes the activation of the inflammasome, to make a long story short. So we have the receptors in an active form in non-infected cells. As soon as there is damage or pathogen infection, there is conformational changes in these receptors, and now there is formation of this beautiful ring, that beautiful 11-fold NLRP3 ring, and the purine domain of the receptors can now be exposed for activation, and they're going to recruit ASC. So ASC is going to be polymerized in the forming these long filaments, and along these filaments there is exposure of the CAR domains of ASC, which now binds to other filaments. So there's a formation of this massive puncta or speck-like structure, in that the inflammasome is going to be robustly activated, and now the formation of these specks or the puncta allows activation of multiple sites for activation of caspase 1, and this is going to recruit the pro-caspase 1, which is produced as a proenzyme in active form, and then it's going to be cleaved and activated in the filaments of ASC, and then leading to activation of caspase 1, and now we have the active form of caspase 1. And of course, caspase 1 cleaves inflammatory cytokines such as IL-1 beta, IL-18, which are produced as a proform, and then it's going to be cleaved by caspase 1. And also another key molecule downstream of the inflammasome is gasdermin D. Gasdermin D is produced also as an inactive form, and is going to be cleaved by caspase 1, and the internal domain of gasdermin D is going to insert this beautiful 27-fold pore in the membrane of the cells, and then this is going to be critical for cell swelling, the loss of the balance between ionic balance between inside and outside of the cells, and the cells going to lyse and promote pyroptosis, which is the lytic cell death, which occurs the release of inflammatory mediators such as IL-1, IL-18, the active caspase 1, and LDH is going to be released as well from the cells that undergo pyroptosis. So this is a very inflammatory form of cell death, which can be triggered by multiple agents, microbes and non-pathogenic agents as well. Then non-infectious agents as well can lead to pyroptosis and inflammation. So this is a picture from a video made many years ago, in that macrophages expressing an LRC4 in fusion with the GFP. So we are seeing the inflammasome responsible for flagellin recognition. And in this experiment, we expressed RFP in Legionella pneumophila, which is a gram-negative bacteria that has flagellin, and flagellin of the Legionella is going to trigger NAPE5, which is going to trigger an LRC4 oligomerization. There's a formation of the ASC spec. So this is a total 2 hours video. 1 hour after infection, we see the formation of this beautiful punta-like structure, which is the ASC in the inflammasome. This is the aggregate of ASC in the cells. And in this region, there is a permeabilization of the membrane by gazemin D, and the PI, which is in the media in this experiment, can go inside the cells and stain the nuclei. So 1 hour after infection, you see the formation of the inflammasome, and 5 minutes later, the cell is completely collapsing, and you see the PI in the nuclei of the cell, and the formation of this bleb, the membrane blebs, and then 10 minutes later, the cell is completely crushed, and then LDH is going to be released, inflammatory cytokine is going to be released, so many signs of the inflammation is going to occur in this process. So this is quite well known for many pathogens, including viral pathogens, but still very unclear about the SARS-CoV-2, how this happens and how this happens in virus. And I'm going to skip the introduction of SARS-CoV-2 in the interest of time, but just to make the point, we had a lot of cases in Brazil. Unfortunately, the situation were not positive, and we did not deal well with the pandemic. We still have a lot of cases. We are only behind US, but if you consider that the American population is much higher than Brazil, we are not in the good situation. And in Ribeirão Preto, the city we are located, we had a lot of cases as well during the last months. So then if you think about inflammasome in response to SARS infection, there was a few papers published before the SARS-CoV-2, which are mainly with SARS-CoV-1, showing that some specific proteins, this is the accessory proteins of SARS-CoV-1, protein ORF3A activates the inflammasome, and this was published January 2019, and I depicted this just to show that, to make the point that this is SARS-CoV-1, not SARS-CoV-2. And this paper was published early last year, and also last year before the SARS-CoV-2, the COVID-19, there was this other observation showing that ORF8b, which is this ORF right here. So a SARS-CoV-1 has a 8A and 8B, so these are two different proteins, and one of them can be cleaved, can activate an LRP3 in response to SARS-CoV-1. And I want to make the point here, this is the SARS-CoV-2 genome, and ORF8b does not exist in SARS-CoV-2 because there is no cleavage of ORF8, so SARS-CoV-2 has only one big ORF8 and not ORF8A and B as SARS-CoV-1. So there's still a lot of unknowns about the fact that if SARS-CoV-2 activates or not the inflammasomes, and the molecular mechanism by which that happens is still very unclear. So if you put in the PubMed inflammasome and COVID-19, I did this yesterday, that is like about 52 results, and most of them are reviews. I think for some reason, people love to write reviews about inflammasomes and SARS-CoV-2. That is really not a definitive paper showing activation of the inflammasome in response to SARS-CoV-2 infection or COVID-19. And if you go through this list, it's going to be a lot of hypotheses, clinical trials, suggestions for treatments, but so on. But so far, there is no convincing data showing that the inflammasome is activated during COVID-19. So we decided to, there was a few papers, clinical data with COVID-19 published early this year from the early patients in Wuhan, then during the first study, I think is the first one that kind of impacted and demonstrated that is a indication of the of cytokine storm in patients infected with SARS-CoV-2, and that suggesting the macrophage activation syndrome associated with severity of the disease. So this study was made with 21 patients in the city of Wuhan in China, and also in Wuhan there was this other paper showing that they enrolled 107 patients, and they were the first one to demonstrate that the LDH is highly correlated with the severity of the disease. So if you remember, LDH is lactate dehydrogenase, is an enzyme that is inside the cells, and as soon as there is pyroptosis or a lytic form of cell death, LDH is going to be released. And they show in this study that the serum of the patients has a lot of LDH in the serum, and this somehow correlates with the severity of the disease. And I guess these two papers launched all this idea that the inflammasome may be very important during COVID-19, and then they foster all these review papers, which kind of important as well, because they show that this pathway may be very important for understanding of the pathophysiology of COVID-19. So, and then we decided to do experiments with the first virus isolated in Brazil, which is a SARS-CoV-2, kindly provided by Edson Dorigon. So we got monocytes from donors, from humans, and then we prime it or not with PAM3C, so just to upregulate the inflammasome genes in the cells. But prime is not really necessary, because only the infection with SARS-CoV-2 is able to trigger cell death in these monocytes, in these primary monocytes. And this happens with or without the priming. Nigiricin is a positive control for inflammasome activation. We see a nice LDH release in response to nigiricin treatment. And then we decided to get the same cells, and we stained the inflammasome intracellularly by using an NLRP3 antibody, and we looked for the puncta, the NLRP3 puncta. Remember, when the inflammasome gets activated, there is the formation of this massive structure with ASC, which is the NLRP3 puncta, and then we can score this in response to infection. We see SARS-CoV-2 infection in primary human macrophages or monocytes. They trigger NLRP3 puncta. This requires the live virus, because the UV inactivated virus fails to induce inflammasome activation. Nigiricin again was used as a positive control, and we see images such as this one. And in this picture, we stained double-stranded RNA. Remember, SARS-CoV-2 is a single-stranded RNA virus, and it happens double-stranded RNA during the virus replication. So this was showing that the virus is indeed replicating inside the monocytes, and we can see puncta of NLRP3 and in this monocyte infected by SARS-CoV-2, which is kind of replicating the side of the cells. So this summarizes a couple of studies we have in vitro showing that the virus can induce inflammasome activation in primary human monocytes and induce the lytic form of cell death, characterized by the presence of LDH, which was supported by the idea, all those reviews showing that the inflammasome may be activated and the LDH may be released due to inflammasome activation in cells infected by SARS-CoV-2. So, and then we decided to work with a group of patients that were enrolled in our hospital and Hospital das Clínicas of Medical School of Ribeirão Preto, University of São Paulo. From April 6th to July 2nd, we enrolled 124 patients. So this is all the clinical data from these patients. They have like comorbidities, laboratory findings, a lot of inflammatory markers, C-reactive protein, D-dimers, LDH, ferritin. Most of them were moderate and severe, because these were patients that looked for the hospital, seeking for the health care in the hospital. So most of them are like moderate and severe form of the disease. And we had 27% death in this group of patients that were enrolled from April 6th to July 2nd, 2020. And by analyzing these patients, we first operate with we measure the active caspase 1. So this is the P20, is the cleaved form of caspase 1, which means active caspase 1, so active inflammasome activation, and IL-18, which is a cytokine that in vivo is specifically cleaved by caspase 1. So the presence of active IL-18 caspase 1 in the serum were measured by ELISA, which indicates the activation of the inflammasome in the patient, which can be measured in the serum of the patients. So this is only data collected at the day of hospital internalization. So the first day the patients enter in the hospital, we collected blood and then measured the inflammasome activation in these patients. So these are controls, and we can see inflammasome is super high in the COVID-19 patients, as shown by caspase 1 cleavage, active IL-18, and as a proof of concept that IL-6 is a known inflammatory cytokine known to be highly abundant in COVID-19 patients. And in our group of patients, we also detect high IL-6. We measure other cytokines by CBA. IL-10 is also high, IL-4 is high, so it's completely dysregulation of the immunity. And interferon gamma is lower as compared to the controls, and no difference. We found no difference between IL-17 and TNF. The fact that interferon gamma is lower, possibly because of cell death of lymphocytes, which is a hallmark of this disease as well. So then we decided to work with the PBMC of COVID-19 patients, and we stained active caspase 1 using the FLICA assay. So the FLICA is a fluorescent dye covalently linked to this little peptide, which binds to the active form of caspase 1. And this is fluorescent. We can measure caspase 1, active caspase 1 by FACS. So this is one example of one control and one COVID-19 patient. So you see that there is activation of the caspase 1 in the PBMCs of the COVID-19 patients. This is just one example, and this is a group of many patients that where we performed this PBMC assay and measure FLICA by FLICA the caspase 1 activation in COVID-19 patients. And then we also looked for immunofluorescence, the activation of caspase 1 by the NLRP3 puncta. So I explained this assay before. We stained the active inflammasome, the puncta of NLRP3, which means the active inflammasome. You can see in magnification of this region of this image, you see the nuclei stained in blue. In the arrow, you can see the NLRP3 inflammasome active in this PBMC of these patients. This is a group of many patients and controls. You see a higher inflammasome puncta formation in patients, in COVID-19 patients as compared to the controls. And we also performed this assay in that we incubated the PBMC cells overnight, and then we used the cell supernatants to measure caspase 1 activation by a luminescent assay. So which means measures active caspase 1 in the supernatants of the cultures overnight. And then we see that in the COVID-19 patients, we see a higher luminescence, which means inflammasome and caspase 1 cleavage in the supernatants of the cells from these patients as compared to the controls. And also many PBMC from many patients induced production of IL-1 beta. This is only spontaneous generation of IL-1 beta, no stimulation with anything, just showing that the PBMCs are infected with the virus and they can activate the inflammasome in the cells as measured by the FLICA, the puncta, NLRP3 puncta, the luminescence for caspase 1 and IL-1 beta. We also got a great help from Professor Alexandre Fabro, which is in the Department of Pathology and Legal Medicine of Medical School in Ribeirão Preto. And there was a major concern about doing autopsy in the fatal cases of COVID-19 because of contamination of the medical personnel. But Professor Alexandre was very well trained to perform this minimally invasive lung autopsy, so they can just sample a little sample of the lung of the patients that deceased due to COVID-19, and then we could have access to these tissues and then evaluate the many parameters of the the infection in the COVID-19 patients, in the autopsy of fatal cases of COVID-19. And in this experiment, in these two pictures, you see the control, you see a nice organization of the lungs as compared to the highly inflamed pulmonary tissues in the COVID-19 patients. This is stained with anti-SARS-CoV-2. You see many infected cells as expected in the lungs of COVID-19 patients. And now we stain it with anti-SARS-CoV-2. And we did this erasing technique that we can stain with many different dyes the same area of the tissue. So we stain it with anti-CD14, and you can see these two cells are CD14 positive, SARS-CoV-2 positive, and they also express NLRP3 in the tissue. Another example is shown in this other image. So you see this CD14 positive cell infected with SARS-CoV-2. You can see even the viral replication inside the cells, and they also express NLRP3, which means the inflammasome is upregulated in the cells that are infected in the lungs of patients that deceased due to COVID-19. But this only shows expression of NLRP3, but not inflammasome activation, which is a confusion in the field, because not always expression of NLRP3 means activation. But the formation of the puncta indeed suggests activation of the inflammasome, and we could find many areas in that we see the cells with the active puncta. So this is a magnification of this region of the image. We see a nice NLRP3 puncta in these cells. This is a macrophage in the lung of the autopsy patient that deceased due to COVID-19. Another example, so this is the inflammasome activation in the cells, and we can score, this is 6 different COVID-19 autopsia, and you see in these patients there was like a higher NLRP3 puncta as compared to controls, which has a very few inflammasome activation. So this is then suggesting that the inflammasome is activated in vitro. It's highly activated in the cells and in the serum of patients, and also in the tissues of the patients that deceased due to COVID-19. We see inflammasome activation in this tissue. And then we decided to get all the clinical data from the patients, 124 patients, and we did this matrix, this correlation matrix, in that we compare multiple parameters, clinical parameters with caspase-1 and IL-18, and you can see that caspase-1 and IL-18 correlate with inflammatory mediators in these patients. Of course, as a proof of concept, caspase-1 P20, so the active caspase-1 in the serum of the patients, correlate with a higher level of IL-18, which is kind of expected. So I think this is a nice proof of concept that our assay is working as expected. But we also see a nice correlation of caspase-1 with IL-6, which is a hallmark of COVID-19. The same thing for IL-18 with IL-6. And inflammatory mediators, we got a very nice correlation between C-reactive protein, which is a marker of inflammation, correlates very well with caspase-1 P20, activation of caspase-1, and active IL-18 in patients. And this is also LDH. As I've shown before, LDH was previously shown to correlate with the severity of COVID-19. And then we can show that caspase-1, activation of caspase-1 positively correlates with a higher level of IL-18, which is somehow raising the hypothesis that caspase-1 may be, an inflammasome may be participating in this lytic form of cell death, which is characterized by high levels of LDH in the patients, in the COVID-19 patients. And now we move on to an association of the clinical outcome with the inflammasome activation in this 124 patients. So first, we separated the patients in the group that received mechanical ventilation versus the group that did not require mechanical ventilation. And we found, not for caspase-1, but for IL-18, we found a higher levels of IL-18 in the sera of COVID-19 patients that require mechanical ventilation, as opposed to patients that did not require mechanical ventilation. And also we got a nice a statistically significant difference in the active caspase-1. When we compared, we grouped the patients by by severe COVID-19 versus mild and moderate. Remember, there was only 7 patients with mild COVID-19. Most of them were moderate and severe, but we could see that the higher inflammasome or caspase-1, active caspase-1 in the patients with severe disease as opposed to moderate and mild. But we did not detect any difference in IL-18. And most importantly, when we separated by the group that did not survive it versus the survivors, we see that the IL-18 was increased in patients that did not survive COVID-19. So this patient deceased to COVID-19, and then we see an increased IL-18 expression at the day of hospitalization. So this is not a longitudinal analysis, but this is only collection at the day of hospitalization. And we found some correlation between poor clinical outcome with increased expression of active inflammasome, caspase-1, and IL-18 in these patients. We did a longitudinal analysis of some patients, and we did not detect differences in caspase-1, but we did detect the difference in IL-18. So this red curves show the non-survivors. So they usually come to the hospital at the early stage of when they enter the hospitalization, they have a higher levels of IL-18 as shown in this graph, and then they reduce during the hospitalization time, but not as low as the patients with which has a mild or a critical and recovered COVID-19, suggesting again that the inflammasome is very important for the clinical outcome of the disease. They are activated in the patients. SARS-CoV-2 can activate these inflammasomes in monocytes from humans, and then they somehow correlate with the clinical outcome of the disease. And this, I'm not going to present this data in the interest of time, but this supported investigations which we are carrying on together with Paulo Lousada and René de Oliveira, which are clinicians that are looking for patients to use therapeutic strategies to inhibit the inflammasome activation in order to treat COVID-19. And they have so far very promising data using colchicine, which is a drug that inhibits microtubule polymerization, and therefore they inhibit as well the inflammasome activation. And these patients has a much better recovery in the hospital, and the results so far is very encouraging. But I'm not going to present this data. This is going to be another story. And just to, there's many people to thank in this story. This is the beautiful campus of the University of São Paulo at Ribeirão Preto. This is the hospital that we work. This is the medical school, is right here. And I usually finish my presentation with a picture of my group, but in this case, I'm gonna finish with all the authors that accounted to development of this work. And I have to give the credit to, especially to Tamara Rodrigues, which is the first author of this manuscript. She's a fantastic PhD student, and she lead all this group of scientists with in the middle of the pandemic with many restrictions. So she did a great job in this study. And also this is only a part of my lab, which worked together with Tamara in this project. And of course, we got help from the Center of Research in Inflammatory Disease, which is the CREED, which is led by Fernando Cunha, which is a fantastic science as well, and we have many other PIs and students. And obviously the people from the clinical, Paulo Lousada and René Oliveira, they're fantastic to collaborate with these guys, and all the help we got to collect the information and grow the patients from all the personnel from the Hospital das Clínicas of Medical School in Ribeirão Preto. And also very important, the help from the virology guys in the Center of Research in Virology, Eurico Arruda, and the students Ronaldo, Ítalo, and Marjorie, as well as the pathology, Alexandre Fabra shown his picture before, Sabrina, Lee, and Mayra, and in the School of Biology, Tiana and Fabio, which helps a lot with the analysis of the longitudinal analysis, especially in the R. And also the help from Fabiane and Ricardo from the pharmacy school, and also in Ribeirão Preto. So all the authors are from University of São Paulo at Ribeirão Preto. So I'm very proud to be part of this beautiful group that work together to tackle important questions in the COVID-19. And of course, more important than everything is the funding, especially from FAPESP, that has providing all this funding to development of this COVID research and other research in the lab. And with that, I would like to thank you guys for the attention and be happy to answer any question if there is.
Thank you, Dario. Very nice presentation and a very good picture of our city of Ribeirão Preto, epidemics in Ribeirão Preto. It's very important for us and for the world too. And Dario, we have a lot of questions here. I'll try to summarize some of them. The first one's from Anderson dos Santos. He's asking if Gasdermin D inhibitors such as disulfirans, if these drugs could have be considered as a therapeutic proposal for COVID-19, and would it be promising to use inflammasome specific inhibitors that would result in decreased processing of IL-1 beta and IL-18, the latter being considered important in the regulation of interferons? I know you've already talked about colchicine, so could you say something else about Gasdermin or any other drugs?
Yeah, so we are setting up like a clinical trial to do the sulfuran. So it's a great question, and this is there's many other clinical trials going on. There's this beautiful paper from Hao Wu and Judy Lieberman showing that the sulfuran specifically binds and inhibits Gasdermin D, and then we are working, and this is led by Fernando Cunha and Paulo Lozada. They're like setting up this clinical trial to test the sulfuran for treatment of COVID-19. So this is ongoing. And the other question is related to the specific inhibitors of inflammasome. Yeah, so one thing I didn't show, and we are kind of investigating this now, is that I think that besides NLRP3, there is other pathway that leads to LDH release. So not all the the cell death is related to NLRP3 activation. So, which means there is other pathways for lytic form of cell death. So the more far away from NLRP3 we get in terms of treatment of COVID-19, I think it's better, and chances of a success in the treatment is going to be better. So if you think about colchicine blocks microtubules, so it blocks many steps during the inflammatory process. And I think this is more promising than like MCC 950, for example, that specific inhibitor of NLRP3. So I think we should go for more downstream, either Gasdermin or either process that blocks many inflammasomes. And we still don't know how SARS-CoV-2 induces this lytic form of cell death. It could be that is necrosis, necroptosis involved as well. And this is all open field to research in the future. And we are somehow addressing these questions as well in different projects in the lab. So these great questions, Anderson. Thank you.
Okay. Another question from Professor José Carlos from the SBI directory. The question is, if the process of activation of inflammasome by SARS-CoV-2 depends on ACE2 receptor, and if the NLP3 activation depends on viral replication, or only the entrance of the virus inside the cells is already able to induce the activation of inflammasome?
Yeah, so this is great questions as well. We never addressed the effect of ACE2 in inflammasome activation, in the NLRP3 inflammasome activation. Monocytes may have a little expression of ACE2, but there is a lot of information about the entries of the virus which are independent of ACE2 as well. We did some preliminary experiments with mouse bone marrow-derived macrophage that does not express human ACE2, and we got some preliminary data suggesting that SARS-CoV-2 can indeed infect these macrophages and can somehow induce activation of the inflammasome. So I would speculate that there is an entry independent of ACE2, which somehow operates, and my idea is that the inflammasome could be activated independent of ACE2. But this is all speculation. We don't have solid data to support this speculation, but our preliminary data suggest that indeed there can be activation of inflammasome independent of ACE2. And the second question is related to the...
If the virus entrance is enough to
replication, yes. So we are doing now a screening of drugs that can block inflammasome activation and virus replication. So we still don't know if the replication is required or not, but my I think it required is the expression of the accessory protein. So I think ORF8, ORF3 may be accounted, so they are pouring, so they punch holes in the cells, right? So then they allow a flux of potassium and therefore NLRP3 activation. So we are addressing this question by just expressing the proteins of SARS-CoV-2 in macrophages and see if they activate the inflammasome to try to especially ORF8. We are very interested in ORF8 because if you think about the genome of SARS-CoV-1, it's very similar to SARS-CoV-2, very, very similar, except for ORF8. ORF8 is very different. It's not cleaved, so it's a completely different protein. And then this can explain a lot of why this virus is so much more pathogenic than the SARS-CoV-1, for instance.
So you have already talked about other possible pathways of the activation, and Karina, our colleague Karina Bortolucci, is asking do you think there are another inflammasomes other than NLRP3 playing a role in the severity of COVID-19?
Yes, Karina, it's a great question as well. I do think there is other inflammasomes activated, and this is how our immune system works, that the beauty of the redundancy of many pathways to sense like whatever pathogenic microbes can replicate in our cells. So I think even without NLRP3, and we may know soon, because we now got the mouse expressing human ACE2, and we are breeding this mouse with NLRP3, and my expectation is that in the absence of NLRP3, we're still going to have inflammasome activation, which suggesting that the NLRP3 maybe it's definitely activated in response to virus, but other inflammasomes may also play a role there. So I think it's
we have two last questions because of our time. We are on time. Lilian Mota is asking you about the viral load. Do you think that the viral load is also important in the severity of COVID-19 besides inflammasome activation?
Yeah, so this is a great question as well. It's interesting that many patients that we have, they are positive for the virus. And I didn't mention that, but all the patients that we use in this study were like PCR positive for the virus. So the viral loads were high in the moment of the infection. But then if you go for after days of hospitalization, the viral loads goes very low, and then starts the inflammatory reaction. So I think it's kind of course they are connected, but the inflammation happens in respect to the viral loads, I guess. So then for some reason, which we don't really understand, the inflammation goes over, and then the patients die many days after the viral loads are low already. So something goes on with inflammation that we cannot still understand and address. But I think to reply Lillian's question, there is indeed like a correlation between viral loads and inflammation. But then I think inflammation is a second step during the disease, and then when the viral loads goes low, then inflammation starts. And then the patient die by the inflammatory process more than the viral loads, I would say.
Okay, and the last question from Professor Lucia Facioli. She's asking you about IL-1 beta. You didn't show any IL-1 beta on plasma serum, I guess. I did not see it. And if you did not detect, I don't know if you did or not detect, but if you didn't detect IL-1 beta on plasma samples, how could you explain the elevated in vitro concentration of this cytokine on the PBMC cultures?
Right. So that's a good question, Lucy. Again, thank you. So the problem of measuring IL-1 beta in the serum is because there is a lot of IL-1 beta produced by neutrophils, and then they can be cleaved by neutrophil elastase and not by the inflammasome. So measuring IL-1 beta in serum, in mouse or in human, it may be misleading because you could be showing something that is neutrophil dependent and not inflammasome dependent. So for this reason, we didn't show any data for in vivo IL-1 beta in the plasma. There is definitely IL-1 beta in the plasma. This is actually published in other papers, but it's hard for us to assume that this is indeed dependent on the inflammasome in vivo. But in vitro, we can measure IL-1 beta in a very convenient way to demonstrate activation of the inflammasome, because we did PBMC, there is no neutrophil. So all the IL-1 beta there is dependent on the inflammasome activation. So, but I would say if you measure IL-1 beta in the serum, we're going to get high levels of IL-1 beta as well. But I like IL-18 better as a readout for inflammasome activation in the patients, because this is specifically cleaved by caspase 1 and inflammasomes.
Okay, so thank you, Dario. And this IL-1 beta in vitro is not after stimulation, so you do not re-stimulate the cells in vitro as you.
No,
as
we
you,
just incubated the PBMC overnight, and yeah, not all patients. If you remember the graph, yes, not all patients have a high levels of IL-1. Some of them does not produce IL-1. So I don't know how is the viral load. So I didn't correlate the viral loads with the inflammasome activation. But in some patients, they do activate caspase 1, and my interpretation is that the cells get activated in the patients, and then they still like producing IL-1 as a result of the inflammasome activation.
Okay, so we are going to finalize here. We are on time. Then there are some other questions. There's one last one. Do we have time for at least one more? From Anderson dos Santos again. So if can ESCRT complexes be considered as a way to escape pyrophytosis and reduce inflammation in lung?
So SCORT, that's it?
I think so. I think so. It's not
okay.
As it's written.
Yes. Yeah, so there's these nice papers from Peter Bros and other research showing that SCORT can somehow inhibit the cell death. Yeah, that's something, it's a nice idea, but it requires clinical tests, and still there's a lot of unknown about the SCORT pathway for inhibition of the cell death. And I don't know the effect of SCORT in the replication of SARS-CoV-2. So before you go to some clinical approaches to foster SCORT activation, then we will need to understand exactly how SCORT would impact the virus replication in the cells. Then it could be a way to go, but that still requires a lot of investigation to support the clinical trial with inhibitors of SCORT, I would say.
And about the IL-18, do you think it could be a predictor of severity and mortality for these patients more than caspase activation, as you showed on the last graphs?
Yeah, I would say C-reactive protein, IL-18, IL-6, LDH, all these inflammatory mediators, they kind of correlate nice with the outcome of the disease. I would say that IL-18 may be one. And there is this beautiful work, I should give the credit to this work, by Akiko Iwasaki, that she did all this longitudinal analysis. This is a Nature paper. And then she also support like IL-18 as an important predictor of the clinical outcome. So that there will be another independent study showing that IL-18 is indeed activated in the patients and can somehow predict the disease outcome, I would say.
Okay, so thank you very much, Dario. Thanks, Amanda, again. And we will finalize this section. And Dario remembered about Akiko, so I'd like to remember everybody that is viewing our transmission via Facebook that Dr. Akiko Iwasaki will be online with us on the next SBI Talks on the October the 8th. She will be talking about the immune response to SARS-CoV-2. So she has very, very important works on the immunity to this virus. And if you could be here to watch again with us, so you are all welcome. So come back on in the next 15 days, okay? So thank you very much. And thank you, Amanda. Thank you, Dario. And we will end our section now.
Thank you.
Thank you.
Thank you.